Okay, so welcome back to this next video in which we are discussing non-homologous end joining in order to repair double-strand breaks within DNA. Okay, right, so we're currently discussing the detection of a double-strand break within your DNA. Okay, so we've discussed that the loose ends of the DNA that have been involved in this double-strand break will get mRN complexes binding to them, which consist of these three proteins, MRE11, RAD50, and NBS1. Okay, then what will happen is ataxia-telangiectasia mutated dimers, which are floating around inactive, they will bind onto the NBS1 proteins. Okay, this then triggers a conformational change in this member of the ataxia-telangiectasia mutated dimer, which is bound to NBS1. It's then going to autophosphorylate a serine residue on itself, basically, and that then is going to completely activate it, and it will detach away from this other clinging on ataxia-telangiectasia mutated enzyme. So this one will come away. Okay, so we've now got this active ataxia-telangiectasia mutated enzyme. Okay, now what is it going to phosphorylate? Well, it's going to phosphorylate a form of histone 2A known as histone 2AX. Okay, but to discuss what that means, I want to just have a little discussion of what histones actually are. Okay, so let's have a little sidetrack to discuss histones. Okay, so firstly, let's just discuss what chromatin is. Okay, so chromatin is a strand which consists of both DNA and protein. Okay, so when people talk about chromatin, what they often draw is something that looks like this. So here is a portion of DNA here, known as the linker DNA, and then what you'll have is a histone here. And then the DNA will wrap around the histone. So it'll go round once, it'll then go round another time, and then it will come off here, and then it will come back off like so, and it will go on to the next histone over here. Okay, so it'll come back off here, and then you'll have the next histone along. So basically, DNA is not just free within the nucleus, it's mainly wrapped around these protein complexes known as histones, basically. Okay, this is what chromatin is, this strand where you have DNA and then occasionally you have a histone which is wrapped around. So it's often compared to beads on a string. Okay, so the histones with the DNA wrapped around them are like the beads. Okay, so here in turquoise, here is a histone with DNA wrapped around it. And then you've got little pieces of DNA between the neighbouring histones, and these are called the linker pieces of DNA, and those are like the strings in between the beads. Okay, right, so what I now want to discuss is the structure of one of these histone cores, as they're called, which are the structures around which the DNA wrap. Okay, in more detail. So let's discuss the structure of a histone core. Okay, so firstly let's discuss the different types of histone. Okay, so there are a huge number of different histones, but they're grouped into 5 families. Okay, so in humans alone there are a huge number of different histones. If you go look in the different species, you know, we've found thousands of these things. Okay, but in humans there are a huge number of different histones, but they're grouped into 5 families. Okay, so there is the H1 family of histones, and the way we group histones into families is that all the H1 family histones will pretty much do the same thing. Then there is the H2A family of histones. Okay, and again there are many different members of this family, but they all do pretty much the same thing. Then we've got the histone 2B family, and I'll stop writing the family now. Then we've got histone 3 family and histone 4 family. Okay, so I want to emphasize the fact that these are not just individual proteins. These are whole families of proteins that all pretty much do the same thing within a histone core. Okay, right, so let's now draw one of these histone cores. So a histone core, which is this structure around which DNA will wrap, consists of 8 separate histone proteins. So let me draw this here. Okay, so I'll draw it as a cube. Okay, so it's going to be an octamer of 8 different histone proteins. Now, 2 of these histone proteins will be histone 2A proteins. Okay, so this one here. 2 of these proteins will be histone 2B proteins. 2 of them will be histone 3 proteins, and 2 will be histone 4 proteins. We'll discuss what histone 1 family histones do later. Okay, right. So let's divide up this histone core into the 8 separate subunits here. Okay, like so. So I'm dividing it up into 8 separate pieces. So we've divided the cube up into 8 separate pieces here. Okay, so let's discuss how we're going to arrange the 2 histone 2As, the 2 histone 2Bs, the 2 histone 3s, and the 2 histone 4s. Okay, so this one here, let's say this one is going to be a histone 4. Okay, so we'll colour it in a certain colour. So this one in blue here, this can be a histone 4. Okay, now the other one then that's going to be histone 4 is going to be this one down here. Okay, like so. So that's also a histone 4 down there. Right, and then these 2, this one here and this one here, these will both be histone 3s. So let's colour those in a specific colour as well. So we'll have histone 3 shown in orange here. Okay, and that one right at the back over there, that will also be a histone 3 protein. Okay, so the histone 3s and the histone 4s assemble into this structure known as the H3H4 tetramer. Okay, so when we consider the full core histone octamer here, it's actually made up of 3 separate structures. Okay, and one of them is going to be this H3H4 tetramer. Okay, so imagine for a moment that these 4, this one here, this one here, this one here, and then the one that you can't see that's right over in the far corner, imagine those aren't there at the moment. If we just have the histone 3 with another histone 3, and then with these 2 histone 4s here, this is known as the H3H4 tetramer. Okay, and this is going to assemble before you assemble up the full octamer. So when you're assembling an octamer, what you take is one of these H3H4 tetramers and you're going to join it onto H2A and H2B dimers, as they're called. Okay, so let's now discuss H2A and H2B dimers. Okay, so this member here is going to be an H2A protein. This member here is going to be an H2B protein. So let's colour these in. So we'll have H2A here in vivid purple. Okay, and then we'll have H2B here in red. So before you assemble the histone octamer here, okay, which consists of the 4 histone proteins, we've discussed that you already have H3H4 tetramers assembled, but you also have H2A, H2B dimers already assembled, which basically consist of a H2A protein with an H2B protein. So this structure that I have coloured in here, so imagine that we haven't got the H3 and the H4 tetramer here, and imagine we haven't got those 2 that haven't been coloured in yet. That then is called an H2A, H2B dimer. Okay, right. Now, when we create the full octamer, we're going to need 2 H2A, H2B dimers. Okay, so the other one is going to be oriented in the opposite direction. So the H2A is going to be facing out this way, and then the H2B is going to be the one that we can't see. Okay, so this is an H2A. So in order to create what's known as the histone core octamer that we've got here, okay, you need 1 H3H4 tetramer and 2 H2A, H2B dimers. Okay, so this is the histone core octamer. Right, so that's the structure of a histone core then. Okay, what we now want to discuss is how DNA is going to wrap around the histone core octamer, and that's the structure that we have within nuclei. Okay, right. And then what we want to discuss is how this is going to be important in the detection of DNA damage. Okay, and the initiation of a response, more importantly, to the DNA damage. So, firstly, how does DNA wrap around the histone octamer like this? So, let's let this green strand represent our DNA. So, it's going to come in here, it's going to go around the outside of this histone 4, then the histone 2b, and it's going to go completely round the back of the histone octamer here. Okay, so it's now gone round the round the histone once. Okay. Now it's going to go round a second time, like so, all the way back around, and then it's going to come out here, and then it will go off. So it wraps around twice. Okay, now let's just, for a complete discussion, discuss what histone 1 proteins do. So unfortunately I've written that exactly where the histone 1 protein should be. Okay, so really the histone 1 protein should be here, so it's going to have to be really thin, like so. Okay, and it's involved in binding to the 2 strands that are coming into the histone and which are about to wrap around the histone, and then the strand which is coming off the histone. So it binds to the 2 and holds them nicely together. Okay, so this is our histone 1 protein here. Okay, so that's the role of all of these 5 families of histones then in wrapping the DNA round like this. Okay, so one more piece of terminology I would like to talk about is nucleosome. Okay, so if we go back to my picture of chromatin now, okay, so here we have a histone here, and then as we know now, the DNA will come in, it'll wrap around twice like so, then it will go off to the next histone core here. Okay, it will then wrap around this one twice, and then it'll go off to the next one. Okay, so a nucleosome is the name for a histone core, okay, with the DNA wrapped around it, along with the linker that takes you on to the next histone core. So this structure we've got here, from the DNA just arriving in onto the histone core, okay, going round the histone core, and then the linker that takes you to the next histone core, that is called a nucleosome. So a nucleosome does not just mean the histone core with the DNA wrapped around it, it also includes the linker strand that takes you from that histone core with the DNA wrapped around it to the next histone core. Okay, so this is the linker strand of DNA. Okay, right. So that's just a nice little bit of terminology to know. Right, so we now want to discuss what relevance does this have for the detection of a double-strand break. Well, we have to remember that where our DNA has broken, we're going to almost certainly be near histones, okay? So the DNA is wrapped around histones. So to put this into context, what I now want to draw is the double-strand break with the DNA wrapped around histones. Now, so that it's a nice picture, I am going to show it as though the double-strand break has occurred in the linker strands. However, the DNA, most of the DNA is actually wrapped around the histone cores. There's much less base pairs in the linker strands than there are within the histone core portions. Okay, so often double-strand breaks do occur in the DNA that is actually wrapped around the histones, but it doesn't make a very nice picture doing that. It makes the picture too difficult. So I'm going to suppose that our double-strand break has occurred within a linker strand. Okay, so let's do this then. So we'll try and draw this picture again. In fact, we could use this picture that I've drawn here. I think I will, in fact. So let's say that our DNA is broken off here. So we've got that loose end here. We've then had the MCN complex come and bind here, which remember consists of those 3 separate proteins, MRE11, okay, then RAD50, and also NBS1, okay. And then what's happened is we've got the recruitment of our ataxia-telangiectasia mutated enzyme here, which remember originally came along in a dimer, but that dimer fell apart once autophosphorylation had occurred. Okay, so there's that phosphate group that's responsible for the autophosphorylation here in blue will show our ataxia-telangiectasia mutated enzyme. So I'll note that up. Okay, so here is ATM in blue. Okay, then we've got NBS1, which I think I'll show in yellow here. Okay, RAD50, which I'll show in turquoise here. And then MRE11, which I'll show in red here. Okay, right. Now, what's going to happen is our ataxia-telangiectasia mutated serine threonine kinase, which is now active, is going to phosphorylate some of the histones which are in these histone cores here, basically. Okay, so it will sort of flap around and bump into histone cores, and it's going to phosphorylate them, basically. Okay, now specifically, it phosphorylates a histone 2A family histone. Okay, and it's specifically histone 2AX. So this is a specific protein now. Okay, this is a specific member of the histone 2A family of histones. So this can function as a histone 2A protein in one of these histone core octamers, but there are many other histone 2A proteins apart from just histone 2AX. So not all histone core complexes will have a histone 2AX in. They will have two histone 2As, but they're not necessarily going to be this specific type of histone 2A. So this stands for histone 2A family, okay, and then it's family member X. So histone 2A family member X. Okay, right. So the ataxia-telangiectasia mutated serine threonine kinase then now is going to phosphorylate serine residues within the histone 2AX. Okay, so let's now show this. I'm not going to show it here though. I'll draw a bigger picture of the histone core complex again. This time we won't show the DNA wrapped around it, we'll just show the histone core complex. Okay, so the ataxia-telangiectasia, which is mutated, is now going to find and then phosphorylate histone 2AXs, which are within histone core complexes. Okay, so once again, here is our picture of a histone core complex here. Okay, and let's now suppose that some of these histone 2A histones are going to actually be histone 2AXs. So remember, this one here and this one here were histone 2As. Okay, so now let's suppose that they are specifically histone 2AXs, or let's suppose that this one specifically is a histone 2AX. Okay, so ataxia-telangiectasia mutated is going to phosphorylate a serine residue within it. Okay, so the single letter code for serine is S. So this is a serine residue here. And I even have the exact number of the serine that is going to be phosphorylated by ataxia-telangiectasia mutated, just in case you actually want to know that. It is 139 that gets phosphorylated by ataxia-telangiectasia mutated. Okay, so here is a phosphate group added on to serine 139 of histone 2AX. Okay, right. So what is now going to happen? Well, once you phosphorylate serine 139 on histone 2AX, it causes conformational changes in histone 2AX that then allow histone 2AX to bind to an enormous great plethora of proteins. Now, we are not going to go through the plethora of different proteins that histone 2AX can bind. We're just going to look at the ones that are brutally essential. Okay, so just to illustrate how many there are here, you know, there is BRCA1, there is SMC1, and many, many others as well. So we're not going to go through them all. We're going to just talk about one here, which is the checkpoint kinase 2. Okay, so CHK2 for short. Okay, and this is also a serine threonine kinase. So this is going to come and bind to the histone 2AX once it's been phosphorylated, along with a plethora of other proteins. Okay, and CHK2 stands for checkpoint kinase 2. So the CH is for checkpoint, and the K is for kinase, and then the 2 is for 2. Okay, so let's colour in the checkpoint kinase 2 in green here. Okay, so this is the checkpoint kinase 2 enzyme here. So it has been recruited onto the histone 2AX that has been phosphorylated. And by the way, there is a special name for histone 2AX once it has actually been phosphorylated on serine 139. Okay, so it changes in conformation so much that it is called something different once it has been phosphorylated. It is called gamma H2AX. Okay, so if you see that, you now know exactly what it means. It means the phosphorylated histone 2AX. Okay, right, which is capable of now recruiting a huge number of different proteins to bind to it. Okay, and I want to stress that the ataxia-telangiectasia mutated enzyme that's active on the end here is not just going to phosphorylate one of these, it's going to phosphorylate any histone 2AX that it comes across in the nearby region, basically. Okay, so there's a nice amplification there. Okay, so we have now got checkpoint kinase 2 recruited to the histone 2AX. So what is going to happen next? Well, basically, ataxia-telangiectasia mutated is now going to just happen to bump into the checkpoint kinase 2 enzyme that's now been recruited here, and it's now going to be phosphorylated by the ataxia-telangiectasia mutated. Okay, so here is that phosphorylation that is put on by ataxia-telangiectasia mutated. And this now is going to activate the checkpoint kinase 2. And now the checkpoint kinase 2 is active. So we've got our ataxia-telangiectasia mutated, which is this active serine threonine kinase. We've now got loads of checkpoint kinase 2 enzymes attached to loads of different H2AX that have been phosphorylated. And those are also active. And what these are now going to do is they're going to activate something very, very important. They're going to activate P53. So the next part of this story goes on to P53. Okay, and we'll discuss P53 and how it's going to be activated by these two serine threonine kinases, ataxia-telangiectasia mutated and the checkpoint kinase 2 in the next video.